Dear students, in this module, I'm going to review the experimental techniques that are employed for the determination of the protein structure. As you know, that there are interactions between the side chains of amino acids that are present in the backbone of the protein. So the greater these interactions are, the more stable a protein becomes, because after the interaction happens, there is a release of energy and the overall energy of the protein molecule decreases. Also, these interactions taken together, they define the overall structure that the protein assumes, and therefore taken together, this information is very useful towards determining the function of the protein as well. As I just mentioned, that these interactions, they define the protein function, then we must know what type of interactions are occurring within the backbone. These interactions mainly occur between polar, hydrophobic, hydrophilic amino acids. And therefore, we need to know which type of amino acid is present at which position in the protein backbone, as well as the 3D structure. Moreover, we need to know when these amino acids interact together. So therefore, for determining the stability, structure, and function, we need to find out the amino acid interactions in the protein backbone. In order to determine the position of these amino acids in the protein backbone, there are several experimental strategies that exist. Namely, X-ray crystallography, as you already know, and NMR. So in this module, I'm only going to talk about X-ray crystallography. In this diagram, you can see the various scales to which our eye can see, that is given by 1 millimeter. If you get hold of a magnifying glass, then you can see up till 10 to the power minus 3 meters, and you can see live and moving objects as well. However, you need to have a microscope, and then you need to have stained samples in order to see up till 10 to the power minus 6 meters. The cost for such a microscope is about 300,000 rupees, and that we cannot see the amino acid side chains using such a microscope still. So for that, we need an X-ray crystallography machine. So the cost for this is very big. It's about 30 million. And that you can see using the X-ray crystallography up to 10 to the power minus 9 meters. So that is approximately 1 nanometer. So you need to have the X-ray crystallography machine in order to study the interactions between the side chains of proteins. Dear students, as I have mentioned earlier, you need to crystallize a molecule and then shine X-rays onto it in order for you to see the positions of atoms within this molecule. So once you know the position of each atom within the protein, then you can construct the 3D conformation of the whole protein as well. Okay, so once you obtain the X-ray crystallography, how does it look like? How do the proteins appear? So here you go. So this is the diffraction pattern that you obtain from the X-ray crystallography machine, and you need to reconstruct the atomic positions by solving the structure, and this is what you see at the end. An important point to note here is that for you to be able to see the positions of each atom, you need to have a resolution that is suitable. In this figure, you can see that the atomic positions are not very clear, and all of these atoms in an aromatic ring, they appear as one single unit. The resolution is about 3 angstroms. However, if the resolution is improved to be 1.2 angstroms, then you can see specific positions for each atom within the aromatic ring. So in order for you to understand the atomic positions properly, you need to have a good resolution, and more so, once you have obtained the atomic positions, then you can measure the distance between various atoms and then talk about their interactions as well. So in conclusion, Upon establishing the atomic positions within the protein backbone, you can enlist the possible interactions that exist between those atoms. So depending upon the distance between the atoms, you can then talk about what type of interaction may be occurring, such as the hydrogen bonds, electrostatic interactions, and others.